Okay, today I'm going to be explaining my answers for the plant biology unit extra credit question. So the question is, do phytochromes, specifically PFR, act like light-sensitive kinases, or does the accumulation of it, meaning the phytochromes, directly cause different mechanisms to trigger flowering genes? So I'm going to first break it down into key terms and points. So first off, with some key terms. First one is kinase. A kinase is an enzyme which is used inside cells and organisms to help regulate complex processes in the cell through phosphorylation, which is the transfer of phosphate groups to substrates to help regulate function. And these are used often as protein kinases, which help regulate genes, specific proteins, or different functions within the cell. Second, there's substrate, which is what I used earlier. Is the substance which is phosphorylated by the kinase, and as I mentioned, it's usually a protein or respective gene that is present inside the cell, and it is the substance that is changed, or its function is changed as a result of the catalytic activity. The third term is Arabidopsis thaliana. It's the plant model organism that is used for genetic plant models. It was used in many of the papers that I'm going to be discussing later. Then there's phytochromes A and B. These are just two types of phytochromes that are mainly only differentiated by their binding protein and their specific mechanisms, and however they both have PR and PFR forms. Finally, there's autophosphorylation, which is pretty self-explanatory. It's just phosphorylation to the kinase itself to help regulate its catalytic function. So the question breaks down into these two sub-questions. At its core, it's pretty much asking, do these phytochromes directly act as kinases, and also, what are the exact mechanisms behind this this activity caused by the phytochromes? So, just to start off, the answer is still uncertain, because there's still a lot of research being done. The full mechanisms are not understood completely. There's been a lot of controversy in the past over what exactly drives phytochromes, what exactly causes the flowering genes to be expressed in a certain way, and there are many theories. However, I'm going to be discussing the theory and argument that it's kinase activity. So, it was first thought of in the 1980s with a paper that discussed how phytochromes could be light-regulated kinases. However, that research died down until around the late 90s, when with the discovery of a cyanobacterial phytochrome CPH1, which is a light-regulated histidine kinase. And this led to other implications that these prokaryotic phytochromes could be light-regulated enzymes, and this showed that maybe it could correlate with plants, could correlate with other things, or these could be like ancestors to present-day plant phytochromes. And also another paper that discusses, that reviews how phytochromes could have ancestry in these histidine kinases, or they could have functions to the different kinases. And this is brought up in later, slightly more present research, where they found different phytochromes, such as the recombinant oat phytochrome A. They found that it acts as a light and chromophore, so it absorbs certain wavelengths of light, and it acts as a light and chromophore modulated protein kinase. And this along with the discovery of 2 phosphorylation sites, which have been mapped for it, shows promising data on the possibility of kinase activity causing the activity of the phytochromes inside plants. There's also in some other reviews how phytochrome phosphorylation in plants have been found and hypothesized, and how phytochrome A and B work in different ways, such as one working in phosphorylation in the cytoplasm, one in the nucleus based on their translocation rate of between moving in and out of the nucleus. However, more research must be done to see how or what exactly these phytochromes specifically do. They also have found that phytochrome B moves into the nucleus in reaction to light, and how phytochrome B is transformed to PRF. And they've also detected lots of kinase activity and autophosphorylation in both the phytochromes, and this brings a lot of evidence and a lot of implications that phytochromes could possibly be a result of kinase activity. Most of these have been recorded in vitro, so inside a laboratory and on a petri dish, not in vivo, which is in life. And we need to test these in the future on these different substrates of the phytochromes, and we need to test it in vivo to see how the functions work in a live plant. So just an overview of the data that I've found. So there's some evidence that phytochromes could possibly act as light or chromophore modulated kinases, and as I mentioned earlier, the discovery of these prokaryotic phytochromes that could be maybe ancestors or this ancestry of histidine kinases. And also there's also evidence in vitro of autophosphorylation and kinase activity in both these phytochromes, A and B. However, in order to find out more research has to be done not only on the kinase activity but also different possible mechanisms. And how we could do this is we could test further substrates in vivo, find more evidence of kinase activity, and try to narrow down what processes exactly affect what, or how these phytochromes specifically act. However, in the future, in order to fully answer this, more research must be done. However, there is still quite a good amount of evidence of the possibility of kinase activity in phytochromes, specifically phytochromes A and B, and this could lead to other research in higher order plants and other plants and how the exact flowering functions work. Here are my references of these 6 papers, and here's a picture of me. Thank you for your time.